Hi everybody, I'm Derrick Pitts, Chief Astronomer at the Franklin Institute Science Museum in Philadelphia, and I'd like to welcome you to a science minute from our group of scientists here at the Franklin Institute. I'm joined by our environmental scientist, Dr. Rachel Valletta, over here on my left, and our Chief Bioscientist, Dr. Jayatri on my right here. So you've probably noticed in the news there's been a lot of talk about measles outbreaks, and it's something that we could have some concern about, but today we're going to talk about measles and genetic therapy, and we're going to take this in a totally different direction than you probably expected we could go with a story about measles. But I promise you, this is going to be a really exciting one that you'll learn a tremendous amount about, and you'll be surprised, your mind will be blown. So I'm going to turn it over to Dr. Jayatri Das to get us started with this, and we'll take it away from there. Jayatri.
All right, well, let's talk about the measles outbreak first, because I think that's what the story that we're seeing in the news this week. There have been over 100 cases of measles here in the U.S. just in 2019. And so one question is, where is it coming from? You might have heard that measles was eliminated in the U.S. Back in 2000. But it turns out that measles is still a big problem in other parts of the world. So what happens often is if there's somebody who travels to one of these other parts of the world, picks up the measles virus and brings it back to the U.S., it has a chance of infecting other people, especially if you end up in pockets where there are lots of people who haven't had the measles vaccine. And that's what's been happening. So you've got some communities in New York, you've got some communities in Washington State, where you have significant numbers of people who haven't had the vaccine. And so the measles is making another appearance.
So I want to jump in there, Jayatri, because we were talking about the difference earlier between eradication and elimination.
Right.
And I think that might fit in somehow here. So can you help us understand what the difference is?
Let's geek out a little bit over some public health terminology and epidemiology. So they sound really similar, but they actually mean two very different things. When organizations like the World Health Organization talk about the elimination of a particular disease, they're talking about the fact that it no longer exists in a certain region of the world, but only in that particular region.
Okay.
When you talk about eradication, that's a whole different scale. That's talking about the fact that this disease no longer exists anywhere in the world. So the best example of a disease that's been eradicated is smallpox. We got rid of it, we don't need to worry about it anymore. We've been trying for years to eradicate polio. We're close, but we're not quite there yet. And so measles is kind of next on the list.
Okay. So you said in the U.S. measles we thought was eliminated. And so as of 2000, you mentioned, we had, is that to say we had 0 cases?
Pretty much.
And now, sort of out of nowhere, we've had this 100 new cases.
And we're seeing this over the last few years because part of the risk is that people forget what these diseases are like, and you sort of think that, well, maybe my child doesn't need this particular vaccine, and so it's a mindset that we call vaccine hesitancy. And as people start to kind of back off from making sure that their kids are getting all of the recommended vaccines, then you have especially kids who are at risk because they're unprotected. And so measles is an incredibly contagious virus. And so it's really important that communities are really maintaining their levels of high vaccination to protect everybody.
So can you tell us a little bit about what the measles virus actually is like?
Yeah, so measles is kind of a pretty nasty disease. And some of the things that make it especially contagious are that it can be transmitted from person to person before the carrier is even displaying any symptoms. So it's not like, once we see the rash, that's when they're infectious. No. It's before that they've already been shedding this virus. And it can last in just air for 2 hours. So if somebody comes to a waiting room in a doctor's office, has measles, but then leaves, and somebody else comes in an hour and a half later, they're still at risk.
So you could find this on a surface, or could you find it as transmitted through the air?
Surface air, absolutely.
Okay.
And that 2 hours sounds like a long time in the air.
It really is. It's shockingly long.
Can you compare it to like the common flu? I mean, does the flu hang out in the air for an hour, 2 hours at a time, or?
So I think probably a better comparison is to a common cold.
Okay.
Because that we think of a common cold as being, really contagious.
Sure.
But in fact, like the cold virus actually needs to be in, some sort of fluid or something to be transmitted.
Okay.
So it can't survive very long on its own in the air. So measles is different in that sense. And what we're starting to learn more and more about is the actual cell biology of how the measles virus interacts with the cells in your body that makes it so powerful and so fast acting.
So why is that? What is unique about the
All
measles
right,
virus
well
in that
let's
regard?
take a look at what the measles virus actually looks like. So when you look at the package that is a measles virus, there's a cell membrane, and on the outer surface of the cell membrane, there are proteins that are kind of sticking out of the membrane. And there are two that are really important. There's one that's called hemagglutinin, and this is kind of a key that the measles virus is just like holding out there looking for the right locks. And then there's a second protein called the fusion protein. So what happens? You inhale the measles virus, it then attaches to immune cells in your lungs, and it's basically using that key to find a lock on your immune cells. And those immune cells then travel to your lymph nodes and travel all through your body. And what they're doing, what the measles virus is doing, is because it's taking over those immune cells, it's suppressing your immune system. So when you get infected by the measles virus, a big risk is that you're now susceptible to a lot of other infections.
Okay.
So there's a bigger risk of getting something
Beyond
like,
the measles.
yes,
Like all
of
kinds
coming
of
down with
others.
pneumonia. In rare cases, but still 1 in 1,000, those measles virus can also transmit to your nerve cells, and that's when you
No
get some like
kidding.
really dangerous symptoms with lasting nerve damage.
Okay.
So it does a number on your body.
So
Yes,
it's
it
that,
sounds it.
So it's that key system that allows it to connect to the immune cells because it's got a key that can go into the lock or connect with the
Right.
lock on the immune cell that will then allow it access to the immune cell to do what it wants to with the cell.
Exactly. So, all cells in your body have proteins on the surface, and it's these interactions that are very specific between the virus and the cells that have evolved because it makes it so much easier for this measles virus to then fuse, that's what the fusion protein is for, to fuse with the immune cell, dump its genetic material into the immune cell, and then basically hijack the immune cell to create more measles virus.
Wow. So it's sort of then this cascading growth.
Exactly. So it's spreading all through your body. But then, this is the particularly diabolical aspect
Okay.
of measles virus, is how it gets itself back out to infect other people.
Yes.
Oh.
Right? So not only does it find a lock on these immune cells, but there's another lock that exists on the cells that line your airway,
Okay.
like your trachea, things like that. And so when these immune cells come into contact with the cells lining your airways, the measles virus finds that second lock on the airway cells. Those cells come into contact, and the immune cells dump the virus into the airway cells. And so the airway cells are then primed. When you sneeze, when you cough, the measles virus goes right back out.
Tricky.
This is really diabolical because it has a way to attack you, but then it also has a way to sort of perpetuate itself and push itself out to attack others.
Exactly. And so this whole cell-to-cell transmission really gives the measles virus 3 advantages. One is that it's fast. So unlike some other viruses that can float around in the bloodstream, the measles virus is right there targeting the cells that it needs to. The second is that it's sneaky. Because the measles virus is hiding inside cells, there aren't other immune cells that are seeing it to be able to pick it up and attack it.
Okay. All right.
And then the third piece is it's incredibly efficient. That because of the specificity of this lock and the key, it's only targeting the cells that it needs to get in, reproduce, and get back out.
Okay. All right. So this diabolical device that can do all this really horrible stuff to it, there actually is a vaccine against this.
There is.
Okay. So now how does
Yeah.
how does the vaccine work against this? And then maybe we can talk a little bit about how else we can use measles to do other things for us in a beneficial manner.
Right. So the vaccine is actually one of the big public health success stories of the 20th century. And it's actually got a great connection to Philadelphia, right where we are. So a scientist named Maurice Hilleman, who worked at Merck Pharmaceutical Company, just a little ways away from here, was the one who discovered the measles vaccine. He actually developed the mumps vaccine as well, using mumps viruses from his own daughter.
Wow. Okay.
And
Close to
then
home there.
combined it with a rubella vaccine that was developed by another scientist named Stanley Plotkin at the Wistar Institute just across town as well.
Sure.
So you may recognize that those three vaccines coming together as the MMR vaccine that every child gets.
Right.
But the way that the measles vaccine works is that it's actually a weakened version of the live virus. So the way that the vaccine is produced is that the virus was infected into chicken eggs and raised over time inside chicken eggs so that it's no longer very well adapted to surviving in humans. So it doesn't do much, when it's injected into the human body as a vaccine, but the structures and the surface of what it looks like is still close enough to the real measles virus that our immune systems, when they see this weakened vaccine, they start generating protective antibodies against it. It's like getting ready. And it has these antibodies in store so that if it ever encounters the real measles virus again, it can match it and destroy it.
Now that process is the same as the process that's used for the way any other vaccine works.
Absolutely.
Okay, great.
Absolutely.
Okay, cool.
But what's interesting about the measles vaccine is that because it's still a living virus, it still actually has a lot of those same structural properties that with the lock and the key that allow it to bind to other cells in your body.
Okay.
And this is where things start to get interesting.
Okay.
So people first noticed that sometimes when cancer patients got infected with measles, their cancer actually got better.
Wow.
Which is weird.
Yes.
Yeah.
Right?
Completely unexpected.
Yeah, this
Yeah,
is not
okay.
something that people necessarily predicted.
Right.
And as we studied what was going on, it turns out that cancer cells also have a particular lock that is a good match to the key on the measles virus.
Amazing.
Okay.
Wow, okay.
All right.
And in fact, one of the locks that the measles vaccine, so the weakened version of the virus recognizes, is a molecule that is particularly prevalent on cancer cells. And so when you have this measles vaccine virus in your body, it's finding these cancer cells in a very specific way. And so this whole, lock and key idea that you can use these matching proteins to target one kind of cell or virus to find another type of cell has opened up, really a whole range of possibilities that I don't think anybody would have ever expected, when you think about what do viruses do. Because we always think about how viruses make us sick. But once we understand how they work, now we can engineer their genetics to make them work for us.
Okay, so now wait, tell us why it's not that we're giving cancer cells measles.
Right.
Okay, because that's what it sounds like. I've got the measles key against the cancer cell lock. Okay,
Yes.
and they match, so that means that we can attach to that.
So we're essentially using the measles virus as a delivery device.
Oh, okay.
So
what are we delivering?
So this is where it's the potential of it is really interesting. So there are right now kind of four different ways that scientists are looking at using the measles vaccine to attack cancer cells. One is to just track where cancer is occurring in the body. And so what you can do is you can take a measles vaccine virus, engineer it so that it can take up iodine.
Okay.
I see where this is going.
You know, inject the virus. You know, the virus goes out and it attaches to all the cancer cells.
And the iodine is the marker.
And then you can add iodine to the bloodstream, and only the measles infected cancer cells will take up the iodine, and then you can use some standard medical imaging techniques like CT scans to look at where the cancer is.
Wow.
So we're delivering radiation in a sense.
Yes. So that's one type of radiation delivery, and that's a very mild form of radiation.
Sure.
You can actually also deliver a more aggressive type of radiation with a different isotope of radiation that will actually kill the cells.
Wow.
So now you've gotten these measles infected cancer cells that can take up iodine. You inject the radiation, and it's specifically taken up by the cancer cells.
Wow. Okay.
And then it destroys those cells.
But again, because it has the right lock and key.
Exactly.
So it's not affecting other cells because they don't have the right combination of lock and key.
And we can even engineer new locks to be even more specific.
Whoa.
Outstanding.
Yeah.
That's on the cancer cells themselves.
Sorry, I got my analogy backwards. We can engineer new keys.
New keys.
To open new locks that already exist.
Okay.
Yes, the locks already exist.
Okay.
We can engineer new keys and specifically put them on the outer surface of the measles virus.
Wow.
So that they are very, very specific to cancer cells, and you can look at particular types of cancers and things like that.
Oh my gosh, that's incredible.
So measles virus on its own, of course, pretty gnarly bad news.
Yeah.
But we've done a lot of amazing biomedical engineering, it sounds, to hijack, in our own right, we're hijacking that virus back. So
We're reclaiming it.
There we go.
This
Yeah.
virus. So this reminds me a lot of Dr. Jim Allison's work. Dr. Jim Allison, of course, is a Nobel winner this year and also a Benjamin Franklin awardee of the life sciences. Can you talk a little bit about Dr. Jim Allison's work and how that compares? Is there any kind of analogous or overlap here?
So they intersect in a really interesting way. So what Jim Allison discovered was that normally when your body it activates its immune system, it automatically shuts it off 4 days later because it doesn't want to overproduce or overreact. But sometimes when you have cancer cells that manage to stick around past the shutdown, those cancer cells grow because your immune system turns off.
Sure.
And so what Allison did was discover molecules that he could use to keep the immune system on, and use the body's own immune system to then continue fighting the cancer. And you know, this led to some remarkable cures for cancers that we haven't seen in any other way before. And this is called the immune checkpoint inhibitors.
Immune checkpoint.
Right, so there are these checkpoints in the immune system that if you can essentially take the brakes off, they keep working.
Okay.
And so where the intersection here is that the measles virus now provides another way of removing those checkpoints. Very specifically in cancer cells.
Okay.
To break up that normal pathway and keep the immune system working.
That's amazing. We can direct our own immune system to behave or not behave in a certain way, in a sense.
Yeah, absolutely. The one that blows my mind the most, though, is that you can even use the measles virus to very specifically deliver chemotherapy.
Okay.
So the idea here is that a lot of chemotherapy drugs can be very toxic to the body, but they're kind of just one step away from a non-toxic version of the drug. And so the idea is that you can administer that precursor to the drug and then use the measles vaccine to deliver the enzyme that will chop off the piece that suddenly activates the drug so that you're only getting that toxic effect inside the cancer cell.
This This is all very cloak and dagger.
I'm having a hard time wrapping my head around this.
It's like
Right.
this hidden suicide scheme.
So wait, this is. Let me see if I have this right. Just a second. This sounds like a version of turducken, kind of, where you've got the duck inside the chicken inside the turkey. But this is a way to sort of like sneakily deliver what you need to deliver right to the right spot without harming anything else around it.
That's the idea. Again, taking advantage of the measles virus's natural ability to spread throughout the body, but designing the keys in such a way that we can control which locks they fit.
Wow. So this has moved beyond the lab, beyond clinical trials. We're actually using these techniques?
It's in clinical trials right now.
Okay.
So there's some good evidence from preclinical trials and animal models. And so there are clinical trials underway right now with certain types of cancer.
Very cool.
So something to look to the horizon for.
Yeah.
But one of the challenges here is that it's even more cloak and dagger because so many of us have been vaccinated for measles.
Sure.
Is that we've actually have to hide the measles virus in order for it to reach the cancer cell. Because otherwise, if you're vaccinated, your immune system will recognize it and try and shut it down before it gets to where it needs to go.
Okay, so it's a turducken within a turducken.
That's what it is.
So,
So it's inside, so that it can work.
So there are scientists who are trying to think about disguising it in inside other carrier cells,
Sure.
or even like covering it in nanoparticles that attach to the outside of the cell but don't interfere with its function.
Wow. That's incredible.
Yeah, I mean, so, we were talking about the fact that biology is really this whole set of chain reactions, that when two molecules interact, it changes shape or it unlocks another molecule that then does its thing. And so what a lot of biology is about, especially molecular biology, is figuring out the chain in which these events occur. And once you figure that out, we can program it essentially to interfere with it in ways that we want to.
That's outstanding.
This must have incredible implications going forward for how to deal with all sorts of sort of like diseases or medical maladies that require certain kinds of medications that don't interfere with other things. Is that what's on the horizon in a sense?
I mean, more broadly speaking, this idea of using the virus to specifically deliver drugs or trackers and things like that is kind of unique. It's this category of therapies called oncolytic virus therapy. Oncolytic means killing cancer cells. But the broader idea of using viruses to deliver genetic material is something that's actually very widely used. And again, we don't always think about this because we think about viruses and how they make us sick. But the key that viruses take their genetic material and stick it into the DNA of their host, that's a technique that's useful for all sorts of genetic engineering. Whether it's specifically for clinical trials or whether it's just for things that scientists are doing in the lab every day. It's almost just just another tool in the toolkit is these deactivated viruses.
Wow. So it sounds like we've known the science on this particular delivery mechanism for some time. Are there any well-known and well-established techniques where we're already using this type of delivery mechanism to deliver some sort of medicine or application or treatment?
This idea of targeted drug delivery is a concept that there's a lot of work around. Viruses are just one way of doing them, but you could create, synthetic cells or synthetic structures that have that same idea, like engineered keys on there. So I would say that the virus is just one example, but whether you're trying to cloak it in gold is another approach to deliver drugs to cancer cells in particular. I think there's a lot of imagination in this space about how we can understand how the human body works and think about the tools that we have at our disposal to really try and fight what is one of the most devastating diseases that we're faced with, because there's so few good solutions to cancer.
Well, it's great to hear that that this is beginning to emerge as something that really can make a difference, and I'm fascinated by this whole area of research about how this can happen and the implications that can come out of it, and I'm sure other people must be really interested in this too. So
it puts measles in a whole new light.
It really does. Yes. Not exactly measles are our friend, but
no.
but a new way to sort of manipulate it to do things for us that we really need to do.
Right.
And to chase that down in a way that really is beneficial for us. That's great. Super. Thanks a lot. So, folks, we've been here today with our Chief Bioscientist, Dr. Jayatri Das, telling us all about how measles can be used in a new and different way to help us with cancer. And also with us is our environmental scientist, Dr. Rachel Valletta. I'm Derrick Pitts, Chief Astronomer at the Franklin Institute. Thanks for joining us, and look for us again soon in the future. See ya.
See ya.